All right, our next talk is by Lea Waller from Charité in Berlin. Unfortunately, she couldn't make it here to Australia, so we have a pre-recording of her talk.
Hi everyone, and welcome to this symposium on harmonized fMRI data analysis with HALFpipe in the ENIGMA consortium. My name is Lea Waller, and I am the lead developer of HALFpipe and the co-chair of the resting state fMRI working group in the ENIGMA consortium. I'm currently based at the Center for AI and Neuroscience at the University of Bonn. So the reason I think this is interesting is that fMRI is one of the few ways that we can use to interrogate the human brain in vivo and discover biomarkers of mental health and psychological states and traits. But the processing of fMRI involves numerous steps to get to usable outputs. That means, unfortunately, that there's been a lot of heterogeneity in how people approach this processing and how they configure each step. And that's really a big problem when you're trying to combine different data sources and achieve larger sample sizes. But achieving those large sample sizes has grown more and more important because there's been a lot of null results and a lot of failed replications in the field of brain imaging. So HALFpipe aims to provide a solution here. HALFpipe enables researchers to agree on an analysis protocol and then run it reproducibly on different data sets and different computing infrastructures. So the way this works is that when you first start HALFpipe, you will see a terminal-based user interface, and that will guide you through data input and configuration. We have recently updated this, and this is the work of the amazing Tomáš Fodran, who's one of the developers on HALFpipe. So when we start up HALFpipe, which we can do with most container runtimes, then it will load the user interface. And the first thing that you're going to be asked is to select a working directory. The working directory is where all of your output files are going to be placed. And I'm just going to select this folder that I've helpfully named working directory. So once I've done that, we can move to the next tab. And you can either do this with your mouse, even though this is a terminal, or use the keyboard shortcuts listed on the bottom. And now that my data can be in vids format, but it doesn't have to be. If it's not, then you just need to tell HALFpipe how your data is structured. But thankfully, the data I'm going to use for this demo, I downloaded from OpenNeuro earlier, so we can just use that and continue. So now that the data is loaded, we are told data successfully loaded, and now we can proceed to the other tabs. So the data input will now tell us how many files it found, and now we can move on to general preprocessing settings, which will have general things and our features. And features are all of the outputs that HALFpipe can calculate. So among those are task-based GLMs. So if you have event files and want to contrast conditions. So for example, let's try one. Let's call that one emo matching. That's one of the tasks that's available in this data set. And we're going to select the emo matching task. I'm going to use both of the experimental conditions that are in this task. And now we can add a contrast. And so now we need to give this one a name, for example, emotion, larger control, and we need to give it some values. So we might have -1 and 1 here. And then for preprocessing settings, this is very flexible, so you can choose your own smoothing, grand mean scaling, temporal filter, and so on. But we generally recommend that you go with the defaults that we have in here. For remove confounds, I usually like to use motion parameters for task-based. But maybe this is not the right approach. Maybe I should actually be using something different. So now I can create a multiverse by clicking duplicate. Now let's create a different one that uses, let's say, ICA aroma. And now that's a copy. And now the only thing I will do is I will go down here, uncheck motion parameters, check ICA aroma, and now suddenly I've set up a multiverse analysis that will let me not just evaluate what's going on in this contrast, but also compare these two processing pipelines. Now the same I can do for seed-based connectivity and network template regression. Those rely on seed masks or network templates. And I can also do atlas-based connectivity. Got to give it a name first. And atlas-based connectivity relies on having an atlas image, and then those that will give you a functional connectivity matrix and also the time series for each of the atlas regions. So now that I've set everything up that I want to run, I can run. So now when I click run, what will happen is that preprocessing will start using fMRI prep. And once a subject has been preprocessed, HALFpipe will immediately start calculating the connectivity matrices and statistical maps that we've just specified. So this will all happen using standardized workflows to ensure reproducibility. Once processing is complete, and this can be either on your local computer or on a high-performance cluster, we ask all users to perform visual quality control. And of course, QC can be time-consuming, which is why we built a browser-based user interface to streamline this. Okay, so here in my browser, I've navigated to the index.html page, which is what HALFpipe generates in the working directory after processing. In this index.html page, at first we'll have a dashboard with three sections. The first one is the NIPipe status, and for every subject, you will be able to see whether processing succeeded or not. And in this case, all subjects succeeded, but if there's errors, then you can also hover over and get the numbers so that you know which participants you need to rerun. So I'm going to hide this section, and in the next one, we see all of the images, and each image that we need to rate is one of these little boxes. And then we have different tabs for all of the different types of images. And so we ask users to rate 6 different images: skull stripping, spatial normalization, TSNR confounds, and spatial normalization. And if any one of those is bad, then the scan will be excluded. And finally, in the bottom, we have a histogram of image quality metrics. So this might help you to look at inconsistencies and also double check participants with outlier values. So each of these dots is clickable and hoverable. So now let's maybe start with subject 1. So we can just click on the box and it will take us to the images. And now I can use either keyboard shortcuts or my mouse to rate these images. I can click to zoom and make sure that all of the brain mask looks good, and then rate them as good, and then continue to the next image. So once you've rated all of these, you can click export. That will generate a JSON file in standardized format that you can either use for HALFpipe group statistics or import into most processing pipelines to exclude the participants that have bad rating. So that means if you proceed with the quality control in this, it's not just going to be useful for HALFpipe, but probably for other studies as well. So these are the 4 steps of HALFpipe processing. And I'm really happy to say that there's been a number of users that have adopted HALFpipe over the years, and that we even have gained some contributors from the community who have sent us code and helped us out with development. Because HALFpipe is an open source project, and we are very much open to contributions from the community. Besides the code, HALFpipe has also been used in a number of really interesting studies. The one that I'm running is a genome-wide association study on task-based fMRI in healthy participants. So we're looking at common genetic variants and their influence on brain activation in emotional phases, working memory, and reward processing tasks. Besides that, other researchers have applied or are in the process of applying HALFpipe to studies of patients versus controls, and they're looking at correlates both for resting state and task-based fMRI for various diagnoses. So I've listed some of these ongoing studies and also some published and preprints over here, and also some posters that might be interesting to check out. So in task-based fMRI, one problem we run into is that every research group generally establishes their own task paradigms. So, for example, on the left is a paradigm that shows you 3 faces, and you have to choose which one on the bottom corresponds to the one on the top. The faces are all showing negative emotions. And this is the experimental condition, and in the control condition, we have the same with geometric shapes. Now, a more recent study adapted this but uses video clips of faces displaying emotions, and you only see one face at the time, and the control condition is also different. So if we now want to do a meta-analysis, how do we combine them? Do we just say, well, everything that shows phases is now our experimental condition, and we put that together in one meta-analysis? And then everything that does not show phases is the control condition. But we could also say, well, all of the phases here are showing negative emotions, so let's only meta-analyze them together with the angry phases from this study. So, and both of these approaches will likely give you different results. And I think this means that it's really important to think about this before starting a study, and to make sure that whatever you meta-analyze in the end makes sense, both in terms of the theory and in terms of what you want to get out of the data later. So both in task-based and resting state fMRI, one problem we have is missing data. And this is not just when we look at tabular data like demographics and questionnaire data, but also brain masks. Because in fMRI, you generally choose the acquisition volume to be quite compact, which helps with voxel size, repetition time, SNR, and so on. But that also means that there's a chance for errors, that the field of view might not be placed perfectly for every participant. And in small data sets, this usually has very small effects. But as data set size increases, as we are aiming to do with HALFpipe, this becomes a major problem, because suddenly now this subset of the voxels where every single participant has data decreases. But we might still want to do statistics on those voxels where not everyone has data. So that means we have this missing value problem, and HALFpipe works around this using a very simple approach called listwise deletion. And the problem here is that this approach assumes that the missingness is completely at random. And this is something that we set out to test. So we asked a few different collaborators to run a logistic regression where the dependent variable is the brain mask, so data or not, and the regressors are demographics. So that means age, sex, and handedness. And the interesting thing is, and this was also surprising to me, is that we see a significant logistic regression in the cerebellum, the frontal cortex, brainstem, and temporal cortex. And so that means we probably need to treat meta-analysis results from those regions with care. So besides questions of missing data and modeling, one problem we run into is the human factor in all of the analysis. So as I mentioned earlier, is that HALFpipe is very configurable, but it comes with recommended settings. And when we set out with the program, we kind of assumed everybody would choose to follow those recommended settings. But interestingly enough, users often choose different settings, either based on personal preference or on the perceived needs of their study or their data. This can create problems because later on we might want to combine their studies into broader meta-analyses that go beyond individual diagnoses or modalities. So to work around this, we decided that we wanted to go beyond a simple recommended settings, but rather have a consensus process to drive these recommendations going forward. And that means Clara Moreau and I created a survey that we sent to researchers across the ENIGMA consortium and also to some experts that we knew and were able to invite. And we asked them which steps are the most important to harmonize. So where do we really need to push for this consensus and push for people to go with the recommendations, and also which settings to use for those steps. So please have a look at her poster that is later today in case you want to learn more. Finally, HALFpipe is a software project. And that means that we have this challenge of shipping a single container file or container that can run all the necessary processing. So this container contains not just the HALFpipe code, but also a copy of fMRI prep, of NILearn, and all the other tools from the open science ecosystem that we use to run all of the analyses. But the problem is that the goal of HALFpipe is that we want to be very reproducible. So whether you ran HALFpipe 5 years ago or last week, we want users to be able to combine those into a meta-analysis. But this is not necessarily the goal of all of our dependencies. So we actually set out to say, let's quantify how much upgrading different dependencies changes our output values. And so the funny thing we found is that, and this was also unexpected to me, is that, for example, when we run fMRI prep multiple times, we don't always get the exact same result. There's a certain stochasticity in numerical calculations always, and this then creates minor variations. So what Violetta did in a really cool study is she ran HALFpipe 100 times on one subject from an open data set, and every time asked it to create a functional connectivity matrix. And so I've just plotted here one exemplary cell from that functional connectivity matrix, and it's a histogram. In turquoise, we see the different runs for the older fMRI prep version, and in orange for a newer fMRI prep version. So now we see those distributions overlap quite a bunch, but it's, for me, really hard to tell whether this is a meaningful difference or whether this could just be attributed to chance. So Violetta came up with this really cool way of quantifying kind of how much energy you would need to move between the distributions, like how big is that difference. And that metric is called the Wasserstein distance, and we can actually use permutation tests to evaluate whether this is a statistically significant change or whether this might have arisen by chance. So in this case, it is not significant. But when we look across the entire connectivity matrix, we do find that for some regions there are statistically significant differences that arise. And so now we know that these are not just random numerical errors that occurred in one version or the other, but we know there's a systematic difference, and we need to go investigate where this comes from. So that's going to be an interesting process going forward, and I hope that we can incorporate that into our development workflow so that we can really ensure reproducibility. Well, thank you so much for listening, and I also want to thank everyone who's been involved in the project. It's been a really amazing collaboration. And in case this talk has made you interested in learning more, please head to our GitHub page. There's also a QR code for that here. And on the GitHub page, we have an issue tracker and a discussion section in case there's any bugs or questions that come up. We also have a README with documentations and all of the download links and commands that you need to get started. We also have a mailing list where we sparingly send emails for new version announcements. And finally, our email addresses from the development team are listed on the ENIGMA website. So in case you have any questions, because unfortunately I cannot be there in person, please reach out via email. Thank you so much.